Hi everyone, in this video we are going to be talking about the steps of meiosis. So we'll start by just remembering what meiosis is and why cells would go through this process, and then we'll get into a little bit more detailed view of what exactly happens in each of the different phases of meiosis. So remember that the end result of meiosis is it's a cell division process that's going to create haploid cells. We used mitosis to create daughter cells that were diploid and genetically identical to the parent cell. But when we go through meiosis, our end result is daughter cells that are only going to have half of the information contained in the parent cell. So what this means is if we're thinking about it in terms of homologous pairs of chromosomes, recall that all of our body cells, so cells like our skin cell, hair cell, heart cell, liver cell, all of our body cells are going to contain 2 of every chromosome. And you saw this when you were doing the karyotype activity. For chromosome number 1, there were a pair of chromosomes. For chromosome number 2, also a pair. So we could say chromosome 1A and chromosome 1B, for example. But these chromosome number 1s are the same size, the same shape, and they have the same genes. So when we're going through meiosis and creating these haploid cells, what this means is that the daughter cells are only going to receive 1 chromosome from each homologous pair. So instead of having 2 versions of chromosome 1, the daughter cells are only going to have 1 version of chromosome 1 version of chromosome 2, all the way through for the 23 sets of chromosomes that human cells have. And the reason that we want to make these haploid cells is that when we're creating gametes, or sperm cells or egg cells, we need to cut that chromosome number in half so that when the sperm and egg combine to create an embryo, that embryo or offspring for whatever kind of organism we're creating is going to have the right number of chromosomes. The full set, but no extras. And this is therefore going to occur in reproductive tissue or germ tissue. Same thing. So this slide is sort of a higher level overview of what we're going to be looking at in a little bit more detail in a minute. So this is showing sort of the big events that are going to happen in meiosis. Like I said, meiosis is starting with a diploid cell. So we're starting with a cell that has homologous pairs of chromosomes, has 2 chromosomes for each 2 chromosomes for each chromosome number. So a set of 2 for each chromosome. So we're looking at homologous pairs of chromosomes in a diploid parent cell. So here we're showing a pair of homologous chromosomes, this green one and this blue one. Maybe we could imagine that the green one is the maternally inherited chromosome and the blue one is the paternally inherited chromosome. The first step is that these chromosomes are going to replicate. So this was also the first step of mitosis as well. So hopefully this feels at least a little bit familiar, that we're going to replicate our chromosomes before we start. And also remember that once chromosomes are replicated, we're going to have 2 identical copies of this same chromosome. So now we're showing 2 copies of the blue chromosomes. And remember that these copies, these identical replicated chromosomes, the 2 sides of it are called sister chromatids. So we've got sister chromatids of this blue one and sister chromatids of this green one. Meiosis as a whole process is going to involve 2 divisions. So there's going to be a first division and then followed by a second division. In the first division, these homologous pairs are going to separate from one another. So we have the 2 chromosomes that are same size, same shape, and have the same genes, but they're going to go to different daughter cells. So the green one is going to separate from the blue one. So we're going to cut the cell down the middle here, and now we have 2 daughter cells, and each daughter cell is only going to contain 1 of those homologous pairs. This first division is sometimes called a reduction division, because you'll notice that at the end here, at this first division, we've gone from a diploid cell that we started with. This cell has 2 chromosomes, 1, 2, and we've ended with cells that are haploid. So this daughter cell only has 1 chromosome, and this daughter cell only has 1 chromosome. That chromosome is still replicated, so you can see it's still composed of 2 sister chromatids, but there's only 1 chromosome here, and there's only 1 chromosome here. So already at the end of this first division, we've created a haploid cell. And then the second division is almost exactly like mitosis. So remember, in mitosis, we separate the sister chromatids into two daughter cells, and that's exactly what's happening in the second division in meiosis. The sister chromatids are going to separate, so we end up with two daughter cells that each contain one unreplicated chromosome. And again, they're staying as haploid because we're not introducing any new genetic material. So the end result is 4 haploid daughter cells. And we'll now look at how this process occurs in a little bit more detail. One thing before we go on to the next slide. So I said that there's 2 divisions that are happening in meiosis, and people usually refer to these 2 stages of meiosis as meiosis 1, which is the first division, and meiosis 2, which is the second division. And another thing, when we go to the next slide, there will be sort of a lot happening, but a lot of the words should hopefully look familiar to you because they're the same stages that we looked at when we talked about mitosis. So prophase, metaphase, anaphase, those words are also going to be used to describe these different pieces of meiosis as well. Okay, so on the slide here, we're showing the different phases that are involved in meiosis 1. So remember, meiosis 1 is just going to be that first division that we're looking at, going from a diploid cell to a haploid cell, or from a diploid parent cell to 2 haploid daughter cells. So we can see here that we've got 2 homologous pairs of chromosomes. There's a larger chromosome here, and a smaller chromosome here and here. So these are going to be our 2 homologous pairs that will follow. And remember that we're going from diploid to haploid. So the weird thing about metaphase in meiosis 1 is that instead of lining up in a single file line, which is what we learned about for metaphase in mitosis, instead these homologous pairs are going to line up 2 by 2. So they're going to line up with their partner chromosome, with their homologous pair. So we can see that the large chromosomes are lined up next to one another, and the smaller chromosomes are also lined up next to one another. And the result of this is that when the spindles attach and these chromosomes go to opposite sides of the cell, we're going to put one member of the homologous pair on this side, to the right side. And same thing with this one. This blue one is going to come to the right side of the cell. And the other member of the homologous pair is going to go to the opposite side. And same thing with this small one. Other small chromosome number 2 is going to go to the left side. So we're separating the homologous pairs from one another and creating daughter cells, because the cell is going to divide right down this middle line, creating daughter cells that have only one piece or one chromosome from the homologous pair. So by the end, you'll see that our 2 daughter cells now only have one large chromosome and one small chromosome. So we've gone from diploid to haploid in our daughter cells by the end of meiosis 1. You'll notice that the chromosomes sort of changed color, and they've got different, like now this chromosome is purple and blue. We'll talk about that in the next video. It's a special process that only happens in meiosis, but don't worry about it for this video. Okay, so meiosis 1 is that first division where we've separated members of the homologous pairs. And then the second part of meiosis is meiosis 2. So you'll see that the end result of meiosis 1, we're starting at the beginning. This is our starting product, or our starting material, our starting cells for meiosis 2. So these 2 cells at the end of meiosis 1 are each going to divide, and there are starting cells for meiosis 2. So you can see these same names, prophase, metaphase, anaphase, telophase, but now we have a 2 after each of them because we're talking about the second division. So this second division is almost exactly like what you're familiar with in mitosis. We're going to divide, we're going to have each of our chromosomes line up in a single file line in the middle of the cell for metaphase. We're going to pull apart our sister chromatids. So here you can see the sister chromatids being pulled apart. And then we're reforming nuclear envelopes and just finishing up by dividing the cytoplasm. So it's nothing new for meiosis 2. It's pretty much exactly what you remember from mitosis. So you can see at the end here, we've got 4 daughter cells, and each of them they're staying haploid because they started off as already being haploid at the beginning, and they stay haploid at the end. And we went through 2 divisions to get those 4 cells. Okay, our last slide in this presentation will just be a quick comparison of mitosis and meiosis. So remember that in mitosis, we're making only two daughter cells. And those two daughter cells are going to be diploid. So they're going to have a full set of genetic information with two copies of each chromosome. And each daughter cell is going to be identical to one another and to the parent. So the goal of mitosis is to preserve that genetic information and keep it exactly the same in the daughter cells. Because we're making two cells, we only go through one division, and this process is used for our body cells, so things like our kidney cells and anything that's not a sperm or egg. In contrast, meiosis, the end product is going to be four daughter cells, and so we're going to need to go through two divisions in order to get these four daughter cells. The daughter cells are not going to be genetically identical to the parent. They're going to have half of the genetic information that the parent cell has. So we're going from diploid to haploid. And there's going to be some genetic differences between the daughter cells themselves and obviously between the parent cell and the daughter cells. In the next video, we'll talk a little bit more about how these genetic differences from these end products of meiosis come about. And like I said, we've got two divisions, and these are going to be used to make sperm cells and egg cells, which are specialized sex cells called gametes. Okay, so in the next video, we're going to be talking about sources of genetic variation in meiosis, and I will see you then.